Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 166th seminar, and I call it a University College London Day. Climate change supercharged the heat wave, leading to death of more than 1,500 people in the European heat wave last week. Also, I heard this week not gonna be different much, while flash floods in Texas, the United States alone led to at least 120 deaths. This is very sad news. One disease can kill one person, but we observe the pandemic could kill more than 7 million of people worldwide. Notably, the climate crisis might eliminate the entire human population. We must take an action right now to provide the next generations with a healthy planet to live in. Hopefully, engineering biology, in collaboration with other fields such as environmental engineering and computer science, will give some solution to this global problem. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Chris Barnes. He is a professor of systems and synthetic biology at University College London, as well as a Turing fellow at the Alan Turing Institute. He is a pioneer who combines computer science with the biology to answer many fundamental questions in cells and microbial community that is very important to translate this fundamental science into potentially real technology that is useful for many, many different areas. Chris, thanks so much for your scientific contributions, and the virtual podium is all yours now, and thank you again.
Oh, thanks for the lovely introduction. I'll just share my slides. So yeah, so it's a pleasure to be here and present the start of this seminar. So I'm going to talk about using AI to engineer biological systems. So we're all familiar in engineering biology, the design, build, test, learn cycle. I stole this nice figure from the paper below. And we should also remember that another factor, if we're going to achieve, the solutions that engineering biology can potentially solve, we should think about scale up as well. This is a big, so we should include this in the design, build, test, learn cycle. So I'm sure you're familiar with this notion of how AI can help. I just want to talk a bit more about that. So really, machine learning or AI, when we talk about AI these days, we're generally talking about machine learning, but it has the potential to help in all areas of this cycle. So we can have new design tools. We can have new ways to program robotics, so that robotics are notoriously difficult to program. So AI, people have developed AI algorithms to help with that. Also, automated experimental design. So this is where we use machine learning and data to design new experiments. And then in the last part, we can also use things like large language models to synthesize knowledge and go beyond traditional statistical inference. So I'm going to talk about two areas: design of design of biological systems, so the design stage, and also the experimental design approach. So this is work that was done recently in my group. So this is using AI to design new genomes. So the idea here is we want to design minimal genomes. So as many people in this field will know, minimal genomes is an interesting area, both from understanding biology, but also for biotechnology reasons. And you know, often streamlining genomes can actually improve production of particular chemicals. This is a nice review that was published recently. And I'm sure you've heard, or if you've not, then I'm sure you will do soon. There's lots of these large language models that are designed for DNA sequences. The most recent and largest being this Evo2 model. And these are they're trained on lots of DNA sequences, and just like a large language model can take in text and predict new text. We can do the same with DNA language models for DNA sequence. However, these are still in their infancy and they're huge and a bit unwieldy. So we've been looking at this idea from a kind of pan-genome gene approach. So just a very quick overview of what we've done is we took this pan-genome in E. coli, and what you can see here is data from around 10,000 different strains of E. coli. You can see that on average, there's about 4,500 to 4,700 genes in each genome. And I'm not sure you can see my pointer. Let's have a look. Over here on the right-hand side, you can see the number of genomes that contain the number of genes. So this represents the core genome and this represents the accessory genome. So many genes only appear in one or two strains. So what we did is we fit a kind of machine learning model, a generative model known as a variational autoencoder. And effectively what we're doing is modeling the presence-absence matrix of these genes. What this particular architecture does is it allows you to learn this latent space. So passes through this bottleneck and it compresses all the data into this layer. We came up with new ways to model the genomes using as small number of genes as possible. And once you've fit the model, what you can do is then sample from it. So we can sample new genomes from the model. And what you can see here is 100 samples of these are synthetic genomes in terms of gene content. And then what we can do is we can convert one of those back into sequence. And that's what we've done here. So these genomes all have around 2,300 genes. So we're effectively halving the size of the genome. And then we can convert those back to sequence. So here you can see the genome size in megabases. So we think this is an interesting approach to design new strains. We're also applying the same kind of approaches to designing plasmids, and I think it's quite a general approach. So this is one way that we can use AI to design new systems. We can also use AI for experimental design. So lots of people have worked on this. You may have heard of active learning. We took a different approach, which uses reinforcement learning, and this is the technology behind the AI that beat the Go champion. This is a really fascinating story, and there's a documentary on Netflix. But basically, reinforcement learning, the agent interacts with an environment through an action and receives a reward. And over time, the agent learns to navigate that environment, be it a game or what we did is apply it to experimental design. So you can imagine we have a system in a bioreactor. And actually, we had another paper that showed how reinforcement learning can be applied to control a microbial community. So in this hypothetical community, there are two species, and the idea is to keep them at a certain level. And the reinforcement learning agent receives information in real time and adjusts these auxotrophic, so this might be amino acids or feed. But what we did is adapt that for optimal experimental design. So in optimal experimental design, we have a model that we wish to parameterize. And, you can look at this cartoon of what a good experiment versus so a good experiment would allow you to infer your model parameters with small error bounds, whereas a poorly designed experiment leaves some more uncertainty in those measurements. So we trained a reinforcement learning agent to design these time course experiments. And what's cool about this is the agent is trained on simulated data before the experiment starts, and then the agent is allowed to control the experiment in order to maximize the data collection. So again, I think this is a nice approach because it combines a black box model, which is the reinforcement learning agent, plus this interpretable mathematical model. And I think, this is a growing approach to automated experimental design, and I think it's going to be it's going to change how we do science. So that's all I want to say. Just to say, if you're interested in the intersection of AI and biological design, then join this interest group that we have. We have people from all over the world. We have a seminar series that will start again in September. So thank you for listening.
Fantastic. Many people now are aware of the importance of the AI or computation. The combining these two, biology and, AI or computer science is very important, and this to solve a lot of fundamental problem, at the same time, some practical problem in the future. So I hope a lot of people kind of adopt both science and then solve a lot of problem in the future.
Yeah, I think there's promise, but there's still a long way to make it really useful.
Sure, absolutely. Thank you.
Okay, thank you.
All right. Now the main speaker of today with a longer introduction. Henry Squire is a PhD candidate in chemical and biomolecular engineering at the University of California, Berkeley, under the advisement of Professor Makita Landry. Actually, she was the keynote speaker last year in the first SynBYSS conference in Hawaii. He is a Department of Defense NDSEG fellow And this fellowship is one of the most competitive fellowships in US. He completed his master's and bachelor's degree in chemical engineering at Case Western Reserve University, where he worked on characterization of complex solvents for grid-scale electrochemical energy storage. Interestingly, Chris also started from physics and then moved into the biology field, and Henry also did same thing from interesting energy storage system all the way to the biology. Prior to starting his PhD, he worked in research and development at Procter and Gamble, one of the big company in the world, where he focused on perfume delivery technologies. In the current lab, his PhD work has focused broadly on developing tools for diverse plant biotechnology applications, including immune priming plants against pathogens and delivering biomolecules to plant cells. Henry, thanks so much for your time today, and please take it away. It's all yours now, and thank you again.
Thank you. I'll share my screen. Let me know
Mhm.
when you can see it. Are you able to see?
Coming up, yes, go ahead.
All right. Thank you. Well, thank you for that introduction and thank you for having me. As was said, I'm Henry. I'm a fifth year PhD student now, and I'll be talking a little bit about some of my work that I've done as a PhD student in Marquita Landry's lab. Specifically, I'll be talking about an interesting discovery that we made sort of on accident of plant-derived cell-penetrating peptides and their use in plant biotechnology applications. So before I get started, I'll give a little overview of broadly what Marquita Landry's lab works on. Broadly, we're interested in using nanomaterials as biotechnology tools. Nanomaterials just referring to any material with a size that is 100 nanometers or less, some aspect ratio that is 100 nanometers or less. Broadly, we use engineered nanomaterials in three biological applications. The first is in imaging of the brain, imaging neurochemicals using nanomaterials as the imaging modality, using nanomaterials for sensing in biological fluids, so sensing glucose in blood, for example, and also using nanomaterials as a delivery tool for delivering biomolecules to plants. I work specifically on this third area of research in Marquita's lab. And under this umbrella of delivery of biomolecules to plants, we're specifically interested in what I call central dogma cargoes. So we're interested in delivering DNA, RNA, or protein to plants, primarily with applications in genetically engineering plants. Our lab has previously worked on delivering DNA and delivering RNA to plants using nanomaterials, focusing primarily on carbon nanotubes. However, recently we've become interested in directly delivering protein itself to plants. And my PhD work has primarily focused on directly delivering proteins to plant cells. We think there might be some advantages to this. One of the advantages in delivering protein, as opposed to delivering DNA, for example, is to avoid some of the regulatory oversight that comes from introducing new DNA to plants, from introducing transgenes to plants. And so broadly, we're interested in direct delivery of proteins for DNA-free modulation of plant physiology, with applications in seed engineering, so developing new varieties of plants, applications in controlling the cellular identity of plant cells, similar to stem cells in animals, or applications in field, for example, to control plant stress. So treating plants with transcription factors to prepare plants for exposure to cold, to drought, or maybe to pests. Now, broadly, while there are a wide variety of tools for delivering proteins directly to animal cells, there aren't very many tools for directly delivering proteins to plant cells. plant cells are anatomically very different from animal cells. I was actually talking with my brother, who's a PhD student, last night, and he was telling me about an experiment he was doing in animal cells where he was doing base editing. And I remember thinking to myself, wow, it's crazy to me that you can so simply do base editing, relatively simple at least, compared to plants, do base editing. Regardless, in plants, we struggle to directly deliver proteins. And so taking inspiration from what's been accomplished on the animal side, where there are a number of different technologies for directly delivering proteins, we've become interested specifically in directly delivering proteins through a technology called cell-penetrating peptides. Now, cell-penetrating peptides are not new. Cell-penetrating peptides, or CPPs for short, I'll use that acronym from now on, were discovered in the late 80s, early 90s. And cell-penetrating peptides are just a chain of amino acids that are capable of directly crossing cellular barriers to cross from outside of a cell to inside of a cell. And you can actually attach cargo to a cell-penetrating peptide, and the cell-penetrating peptide will take the cargo with it across these cellular barriers. Like I said, these were discovered in the late 80s, early 90s, and they're really well studied in animals. There's about 10,000 papers on the topic of cell-penetrating peptides in animals, and there's over 2,000 or approximately 2,000 unique cell-penetrating peptide sequences that have been reported. A cell-penetrating peptide sequence just referring to the specific amino acids that make up the cell-penetrating peptide. Now, while CPPs are really well studied in animals, they're not well studied at all in plants, and a systematic study of CPPs is currently missing. There's a handful of papers on cell-penetrating peptides in plants, less than 100 by my reading of the literature. And I'd say that there's really no widespread application of cell-penetrating peptides in any sense in plants. And I'd say that one of the challenges we faced in the field initially was it wasn't entirely obvious if cell-penetrating peptides actually function in plants because there was a lack of a good tool for studying cell-penetrating peptides in plants. More specifically, it was difficult to evaluate the fate of a cell-penetrating peptide in a plant. And when I say fate, I mean it wasn't obvious when we treat a plant with a CPP, where does the CPP actually end up? Does it actually successfully enter the plant cell, or does it get stuck somewhere along the path between outside the cell and inside the cell? Historically, the way to study this was a technique called colocalization analysis, where you would take a reporter plant that expresses some cytosolic reporter like green fluorescent protein, so the cytosol of the plant, the inside of the plant cell glows green. You would treat that plant with a fluorophore tagged peptide that fluoresces a different color, like red, for example. You would image the plant and identify the location of the cytosol. So this is the location where you're trying to deliver your cargo. Then you would image the fluorophore tagged peptide that you treated the plant with, which fluoresces a different color. And you would overlay these two images and look for areas where they overlap. And that would suggest to you where if delivery had been successful or not. With the idea being that if the red signal overlaps with the green signal, then that would indicate successful delivery. Now, for those who are familiar with looking at microscopy images of animals, they might look at this green jigsaw puzzle looking shape here and think that's odd. This is the cytosol of the plant. The plant, again, has a very odd cellular anatomy relative to animals, where the cytosol of the plant is compressed right up against the cell membrane and cell wall by the large central vacuole that's in the plant. And so there's only a very thin band of cytosol with which you can image against. And so this creates a challenge. There are actually a variety of different fates that a cell-penetrating peptide can experience on the path to successful delivery. And so the technique that I described on the previous slide actually doesn't have sufficient spatial resolution to differentiate between all these different fates that a cell-penetrating peptide can experience. So on the left side, I have a cartoon image of a red fluorescent cargo of some sort that we're attempting to deliver to the cytosol of a plant that's marked in with some green fluorescent object. On this object's path to inside the cytosol, there's a variety of different fates that the cargo may experience. The first is that the cargo might be too large to cross the cell wall. The cell wall is a barrier that's unique to plants. Animals do not have this barrier. And the cell wall in plants is highly exclusive. The size exclusion limit is about 5 to 20 nanometers. So any objects that are larger than that size exclusion limit simply are not going to cross the cell wall without applying external force. Another fate that a cargo could experience on its way to the plant cytosol is entrainment. The cell wall actually has non-negligible thickness, and so there are objects can become stuck within the thickness of the cell wall, particularly if they're on the similar size order to the size exclusion limit of the cell wall. Another potential fate that a cell-penetrating peptide could experience is sequestration. So if a cargo and its cell-penetrating peptide successfully cross the cell wall and maybe even start crossing the plasma membrane, it may become sequestered non-functionally to an endosome or to a lysosome if the cargo is endocytosed, for example. This would appear to be in the cytosol, but it's not actually functionally within the cytosol. It's been sequestered to a non-biologically functional area. Another potential fate, maybe the final fate that I'll discuss here that we're interested in, this is the one that we actually want to achieve. This would be successful delivery, so where the cargo actually reaches the cytosol. And so all of these different fates, all these different possible fates for a cell-penetrating peptide and its cargo occur on a length scale of about 200 nanometers, meaning the space between the cytosol and outside of the cell wall is about 200 nanometers. And standard confocal microscopy does not have sufficient spatial resolution to differentiate between all of these different fates. We wouldn't really be able to tell the difference between entrainment and delivery. You especially wouldn't really be able to tell the difference between sequestration and delivery. And so, in our view of the literature, it wasn't entirely obvious if cell-penetrating peptides are even functional in plants because colocalization analysis, the method that I described on the previous slide, was ambiguous. It didn't have enough spatial resolution to tell what was actually happening. So to solve this problem, my mentor in the lab, my previous grad student, started working on this reporter system that's based on the principle of split GFP fluorescence. So GFP, green fluorescent protein, can be split into two pieces, neither of which are fluorescent on their own. However, if these two pieces are in the same biological compartment or the same biological location, they will spontaneously complement and reconstitute GFP fluorescence. You'll be able to see green fluorescence coming from the protein at that point. We took this principle and created a reporter system that would allow us to evaluate if a cell-penetrating peptide works in plants or not. We call the system Delivered Complementation Implanta, or DCIP. That's the name for the system. And the system works pretty simply based off a reporter protein that consists of a fusion protein of M. cherry, a red fluorescent protein, fused to a broken GFP protein, so GFP 110. This construct is fused to a nuclear localization signal, and so this construct is localized to the nucleus of the plant. To screen if a cell-penetrating peptide functions in a plant, we tag a cell-penetrating peptide with GFP11, the other component to split GFP, and treat a plant that is expressing the reporter system with this fusion peptide. If the peptide successfully reaches the cytosol and the nucleus, then what we'll observe in the plant is reconstitution of green fluorescence. So we'll observe GFP fluorescence. This allows us to differentiate between entrainment or exclusion or sequestration because we will only observe green fluorescence if and only if successful delivery occurs. So more practically, the way that we use this system, we work in the model plant Nicotiana benthamiana. It's a relative of tobacco, and it's the equivalent of the mouse in the plant world. It's one of the model plants that we work with. We force this model plant to express the DCIP reporter system that I described on the previous slide. And if you were to look on a microscope at the plant that is expressing this reporter system, what you would observe are red fluorescent dots, which are the nuclei of the plant. And this would be the plant expressing the reporter system. You would only see red fluorescence, though. You wouldn't observe any green fluorescence because, remember, we split the GFP. Once the plant is expressing this reporter system, we would then treat a plant with a fusion of GFP11, the other component of GFP, fused to a cell-penetrating peptide that we're interested in evaluating its function. After treating the plant with the cell-penetrating peptide fused to GFP11, we would go down to a microscope and we would observe the plant again under a microscope. And in cells which we have successfully delivered GFP11, we would observe reconstitution of GFP fluorescence. So in these cells, we would observe overlapping both red and green fluorescence. We would then put the images through a computer process. The computer would count the number of red fluorescent nuclei and the number of green fluorescent nuclei, and the ratio of the number of green fluorescent nuclei to the number of red fluorescent nuclei then represents the cellular delivery efficiency. And so the reason we nuclear localize this whole construct is to simplify the process of counting nuclei such that we can get a delivery efficiency on a cellular basis as the output from this assay. And so hypothetically, what the DSIP workflow is allowing us to do is it gives us an unambiguous quantitative measure of cell-penetrating peptide performance in plants. So I'll demonstrate how it works. We first sought to investigate if DSIP was functioning by working with the model cell-penetrating peptide R9. R9 is really well studied in animals. It's one of the most efficient cell-penetrating peptides. It's just a cell-penetrating peptide composed of 9 arginine amino acids back to back. We fused R9 to GFP11, and in total that was 3.5 kilodaltons, so not too large. We then treated plants that were expressing the DSIP system, and what we observed when we looked for green fluorescence is green fluorescent nuclei. So these green objects that I'm showing you here are nuclei that have green fluorescence that has been reconstituted due to successful delivery of GFP11. So we're able to visualize successful delivery events. When we flip over and look at the red fluorescent nuclei, so these are all the possible nuclei we could deliver to, we see that the nuclei that we did successfully deliver to co-localize with the nuclei that express M-cherry, so marked in or express the red fluorescent protein. Marked with white arrows, these are nuclei that we've successfully delivered to. Then on the far right, you'll see 2 examples. These are cells which we did not successfully deliver to, marked in red. And so we can count the total number of red nuclei. We can count the total number of green nuclei. And then that gives us this quantitative metric here, like I'm showing you. And we can do this for a variety of different concentrations of R9 and GFP11. And as we increase the concentration, we see that delivery efficiency increases up to about at 100 micromolar of this construct, we achieve a delivery efficiency of about 50%. So 50% of cells we successfully delivered to. Next, we did a control experiment, an important experiment. We removed the R9 cell-penetrating peptide, and we investigated, does GFP11 on its own deliver itself to the plant cell? And what we find is no, GFP11 requires a delivery tool to successfully enter the cell, which makes sense. The reason why we have the field of biomolecule delivery is because biomolecules of their own accord do not typically go where you want them to go within the plant. You have to design a tool that takes the biomolecule of interest to the biological location that it needs to be. So that was not surprising that GFP11 on its own does not successfully enter the plant cells. Now, what I've shown you is interesting in the sense that we've demonstrated that we can visualize CPP-mediated delivery events in plants, but this is just a toy example in the sense that we haven't delivered a full-length protein. We've only delivered a small peptide, GFP11, and quite small at that. The whole construct is about 3.5 kilodaltons, which we want to deliver something that can actually do interesting biology. So next, what we sought to do was to deliver a full-length transcription factor. We sought to deliver specifically a protein called WUSCHL. WUSCHL is a protein that's involved in developmental regulation in plants and maintaining plant stemness, and it has some applications in plant biotechnology for genetically engineering plants. So we expressed a Wuschel protein that has a R9 cell-penetrating peptide tag to attempt to deliver it, as well as a GFP11 tag to track the internal location of the Wuschel. And in total, this construct was 42 kilodaltons, or about 10 times the size of what we were delivering previously. When we treated a plant that was expressing the DSIP reporter system with this construct, what we observed was about 50% of cells we were able to successfully deliver this full-length protein to about 50% of cells. So that was a pretty exciting result for us. In our minds, this was one of the first demonstrations, unambiguous demonstrations of delivery of a full-length protein to plant cells using a cell-penetrating peptide. Obviously, this doesn't demonstrate that the Wuschel is functional. It just demonstrates that we have put the protein inside of the plant cell. There were separate experiments that we needed to do to evaluate the functionality of the Wuschel. What I want to actually talk about today, though, is when we conducted the same control experiment that I illustrated on the previous slide by removing the R9 cell-penetrating peptide, we unexpectedly found that Wuschel was somehow delivering itself. We would expect that when we remove the R9 cell-penetrating peptide, that Wuschel would no longer enter the plant cells. But we were really surprised to see that Wuschel was still entering plant cells at the same efficiency as when it had a cell-penetrating peptide. This was a really surprising result to us because, again, GFP11 on its own does not internalize into plant cells, but somehow Wuschel, which is 10 times the size, was. And so we were quite perplexed by this. We went back to literature, and Wuschel is actually a part of a class of protein called homeoproteins. Homeoproteins are transcription factors that are involved in developmental regulation, and they're broadly conserved across eukaryotes. And homeoproteins were actually the source of the second ever discovered cell-penetrating peptides. Specifically, the second ever discovered cell-penetrating peptide was discovered in a homeoprotein called Antanapedia while studying fruit flies back in the 80s. And Antanapedia is a homeoprotein, like I said. And to be classified as a homeoprotein, a protein must contain a domain, a 60 amino acid domain, which has a characteristic 3 alpha helices back to back, in a very characteristic structure. What's interesting about Antanapedia is the third alpha helix of Antanapedia was discovered to be cell-penetrating. And this was the second ever discovered cell-penetrating peptide. It was discovered somewhat on accident. And what's interesting about this is generally since the discovery of the cell-penetratingness of the third alpha helix of Antanapedia, animal homeoproteins in general have been discovered to be generally cell-penetrating. Most animals have multiple different homeoproteins, hundreds on average. And in general, if you choose a random protein from the homeoproteins that are within a specific animal species, in general, you'll find that homeoprotein is cell-penetrating. Specifically, the third alpha helix is cell-penetrating. So what's interesting to us about that is Wuschel is also a homeoprotein. It's a plant homeoprotein. Wuschel is involved in developmental regulation. It specifically regulates development of the meristem and maintenance of the meristem in plants. And if you align the third alpha helix of Wuschel with the third alpha helix of Antanapedia, so align the amino acids that make up the third alpha helices, you'll find that the third alpha helix of Antanapedia, remember this was the second ever discovered cell-penetrating peptide, looks very similar to the third alpha helix of Wuschel. And so this suggested to us that potentially the third alpha helix of Wuschel might be cell-penetrating to plants. That might explain that weird result that we saw a couple slides ago, where Wuschel was delivering itself somehow to the plant. Furthermore, this led us to the hypothesis that maybe plant homeoproteins in general are cell-penetrating, and they could be an interesting source of cell-penetrating peptides for plants in general that might already be optimized for plant applications by evolution, hypothetically. So to probe this hypothesis, we first investigated how many homeoproteins could we actually use as templates for new cell-penetrating peptides, specifically derived from plants. Well, there are two sources that we looked at. There was a review paper from 2009 which cataloged plant homeoproteins. and it cataloged about 812 different plant homeoproteins. Since 2009, there's obviously been an explosion in sequencing technology in terms of the number of plants that have been sequenced and just our ability to sequence. And so I also consulted a newer database called UniProt, which had over 200,000 different plant homeoproteins cataloged. There was a lot of redundancy across these two sources. Even within the UniProt database, there's a lot of redundancy. And so after doing a quality check, essentially what I found was there was about 40,000 unique plant homeoproteins, which could serve as a template for cell-penetrating peptides. So after identifying the possible homeoproteins which could serve as a source for cell-penetrating peptides, the next step was to actually extract the specific third alpha helix peptide sequence from each of those homeoproteins. So using Clustal Omega, we simply aligned all of the homeoproteins and identified the third alpha helices and extracted those third alpha helices from those homeoproteins, finding that there were 3,500 unique cell-penetrating peptides making up the third alpha helix of the 40,000 different plant homeoproteins that we found. This was interesting to us because there's obviously a lot of redundancy, only 3,500 unique sequences out of 40,000 possible. Broadly, there are 20 natural amino acids. And so hypothetically, there were 20 to the 17th possible different sequences. And so obviously, given that there was so much redundancy, evolution has constrained, unsurprisingly, the design space for third alpha helices quite significantly for us. And so in our mind, evolution has maybe done part of the work in constraining what could be the design space for third alpha helix peptides. We're next curious if there's a specific structure to the design space. How were the third alpha helix peptides related to each other? So we created a matrix representation of the peptides using BLOSUM 62 substitution, and then conducted dimensionality reduction using principal component analysis. And what we find is that there's not really any obvious structure to the reduced dimensionality representation of the peptides. We find that, there's not really any obvious structure that would indicate how we should select peptides from this space. And so we somewhat randomly chose 30 peptides across this design space and chose those to do experiments. I can't do experiments on 3,500 different peptides, and so we chose a subset of 30 from across this space to do investigations with, to investigate experimentally for their cell penetratingness. Here are the 30 different sequences that we ended up selecting. On the first, I'll point out on the top, this sequence that I've labeled AT whoosh. This sequence is the third alpha helix derived from whooshel. And so this was sort of the source of our original investigation. All of these sequences on the left are derived specifically from the model plant Arabidopsis. Arabidopsis has the best annotated genome, and so it has the best source of proteins for us to investigate. On the right, these are all sequences derived from a diverse set of species, ranging from corn to moss to furs to algae. And then the last sequence I'll point out is a sequence that I've called random. This is a sequence that is not a plant homeoprotein, but was a random protein that we aligned, another transcription factor that we aligned along with the homeoproteins that we aligned, and extracted the portion that aligned just to investigate if if other proteins generally have this property of being cell penetrating, and if a random protein shouldn't have this property. And so this was a control for our purposes. So next we took the Arabidopsis whooshel sequence and tagged it to GFP11 to investigate if a third alpha helix derived from a plant homeoprotein is cell penetrating. We used the DSIP system that I described earlier, and we treated a plant with this peptide construct. And I'm not sure how well you can see it on the screen, but there are 3 nuclei here, which are green. So these are 3 successful delivery events mediated by a third alpha helix peptide derived from a plant homeoprotein. Obviously, these overlay with the red fluorescent protein M cherry, and in white I've marked these successful delivery events. And so this was a preliminary demonstration to us that third alpha helices derived, at least from Wuschel, that sequence was cell-penetrating. We repeated this experiment with all of the different peptides that I showed you on the last slide with all 30 sequences. And when we conduct that experiment and quantify all of the images, this is what the data looks like. And so generally what we find at a high level is that the third alpha helix of plant homeoproteins are generally cell-penetrating. So this is mirroring a previously known capability of animal homeoproteins. So that's an interesting bit of biology to start with. Second, what you'll see is that there's really wide variation in the cell-penetrating efficiency of these peptides, varying from just at 30% over here on the left, down to peptides which really don't work at all. And so that was quite interesting to us. Marked in dark blue here is R9. This was currently the most efficient cell-penetrating peptide in plants. And what we found with, I guess, no design rule other than looking for sequence diversity, we've already found 5 examples which perform on average better than R9, which implies to us that somewhere in the space of 3,500 different cell-penetrating peptides derived that could come from plant homeoproteins, potentially there's one that's quite efficient and even more efficient than the ones that we've already identified here. Because again, we just identified these simply by luck. So that was interesting to us. Perhaps this isn't that impressive though, in the sense that the delivery efficiency is a bit low. 30% isn't that high yet. And so we next sought to investigate the concentration dependence of delivery efficiency. So what we demonstrated was that as you increase the concentration of the peptides that you treat the plant with, you'll see that the delivery efficiency increases to about 70-ish% at 200 micromolar. And so this demonstrates to us that we can control delivery efficiency just by modifying the concentration. Something I'll point out here is that relative to animal systems, these concentrations are quite high. And so that's something that we're interested in investigating in the future is why is it that it requires such a higher concentration of cell-penetrating peptide to achieve successful delivery events in plants relative to what we observe in animals? We can do some statistical tests on which of these delivery efficiencies are statistically different. I won't go into detail about that for now. But in general, what we're observing is that the peptide that I call CL-WOX, the most efficient peptide that I showed you on the previous slide, this peptide is outperforming R9 at a variety of different concentrations. And the peptide that I call ATDDT, this peptide doesn't really seem to function significantly different from the mock treatment at any concentration. And so there's some explanation for why certain peptides derived from third alpha helices are functioning, whereas others are not. And that requires some more investigation in the future. So we were interested in understanding why this peptide CL-WOX is so efficient relative to other peptides. And so we're specifically interested in understanding which amino acid making up this peptide might be the most important, or which set of amino acids might be important. So we systematically mutated the amino acids that make up this peptide, starting from the left-hand side all the way to the right-hand side, systematically mutating one amino acid at a time with an alanine, and in the end, attempting to understand which of these amino acid residues are critical to enable internalization of this peptide. So when we conducted that experiment, what we found were, in general, mutating any of these core amino acids would reduce internalization efficiency. So specifically mutating position 3 reduced delivery efficiency by about half. Mutating this tryptophan at position 6, which is really conserved across all of the different peptides that we investigated, that reduced delivery efficiency as well. Removing any of the positive charge at the C terminus of the peptide also reduced delivery efficiency. And so in general, what we're finding is that there's an interesting balance of hydrophobicity and charge that's necessary for the delivery efficiency of this peptide, which sort of matches what was previously reported in the animal literature. In general, I'd say that we haven't made much progress on really determining why a certain peptide from this library works, whereas another peptide does not. And that's going to be an area of future investigation for us. In general, we found that for peptides that are very similar in the library that I showed you on the previous slides, even a single amino acid mutation can drastically reduce delivery efficiency, taking the peptide from being one of the top performers down to a peptide that doesn't work at all. And the mutations that cause this to happen are often non-intuitive. We don't have a good explanation for and a good physical intuition on why that particular mutation would cause that particular drop in performance. And so this is going to be an area of future investigation for us. We haven't made much we haven't made much ground on that front yet. Moving forward, we were also curious about how exactly are these peptides internalizing into the plant. We didn't have a sense for the mechanism by which they were functioning. In animal literature, there's broadly two postulated mechanisms by which cell-penetrating peptides cross cell membranes. Obviously, in plants, peptides have to cross both a cell wall and a cell membrane. But in animals, it's postulated that there's an energy-independent mechanism by which peptides transiently disrupt the membrane and directly cross the cell membrane to enter the cytosol. There's also an energy-dependent mechanism where the cell-penetrating peptide induces endocytosis by some mechanism and enters the cytosol via endocytosis. To attempt to investigate which one of these mechanisms might be playing a role in plants for our peptides, we pre-treated a plant with wortmannin, which is an endocytosis inhibitor, and then conducted the same DSIP assays that I've been describing, where we treated the plant with GFP11 fused to CL-WOX and conducted a DSIP assay. When we do that experiment, what we find is independent of concentration, plants that are pre-treated with wortmannin and thus have suppressed endocytosis have suppressed delivery efficiency of the CL-WOX peptide, which suggests to us that endocytosis is playing a role in the successful delivery of cell-penetrating peptides. However, I'll note that we don't see total abolition of delivery, which suggests to us that there's a mix of mechanisms happening, both energy-independent and energy-dependent mechanisms, which sort of matches animal literature, where depending on the specific cell-penetrating peptide, the concentration of the peptide, and the type of cargo that the peptide is carrying, the mechanism of entry into the cell varies. So perhaps what I've showed you so far, you recall that I motivated this talk by explaining that we're ultimately interested in delivering functional protein cargoes. And so to show you that we're actually moving in that direction, towards applications in seed engineering, cellular identity modulation, and stress protection, we demonstrated that we can deliver a protein called Cre recombinase using cell-penetrating peptides. Cre recombinase is a protein that can conduct recombination on sections of DNA at sites, around sites that are called LOXP sites. Cre recombinase recognizes LOXP sites, performs recombination, and can remove the sequence of DNA that is in between these two LOXP sites. Practically, this is used in plant genetic engineering to remove selectable markers that you do not want in your final engineered plant. And so there are some practical applications of delivery of Cre recombinase, but for our purposes, we attempt to deliver Cre recombinase because it's easy to assess if Cre recombinase is functional or not. So we developed this reporter system that consists of two transcriptional units. The first transcriptional unit consists of a red fluorescent protein M-cherry that's driven by its own promoter and terminator. On the second transcriptional unit, there's a second system that drives expression of GFP. However, this system is blocked by a premature terminator that's flanked by two LOXP sites. What this system allows us to do is, in plants that are expressing this reporter system, what we would observe is red fluorescence, but we would not observe any green fluorescence. When we treat the plant with Cre recombinase, if the Cre recombinase successfully enters the plant cell, and if it's functional, then the Cre recombinase will recognize the LOXP sites and excise the premature terminator that's in between the two LOXP sites. what this would enable is this would turn on both red and green fluorescence. So plant cells that we've successfully delivered Cre recombinase to, we would observe both red and green fluorescence. And so in a similar manner to DSIP, we can then count the number of red and green cells and get a sense for the delivery efficiency of Cre recombinase. So we express Cre recombinase tagged to the CL-WOX cell-penetrating peptide that we previously discovered. This construct is 44 kilodaltons in size. And when we treat a plant that is expressing that reporter system with this Cre recombinase tagged to CL-WOX, what we observe is reconstitution of GFP fluorescence. So what you're seeing here is GFP that's been turned on by successful delivery of Cre recombinase. And you'll see that it overlays with M-cherry with red fluorescence. When we quantify these images and run the control experiment where we remove the cell-penetrating peptide, in this specific instance, we find that Cre on its own does not successfully deliver itself, and we find that we're able to deliver Cre recombinase functionally to about 20% of cells. And so compared to current methods of delivering proteins to plant cells, we're about an order of magnitude more efficient. And so we're happy with that. We also think that's an improvement. We obviously would like to do better. And so there's future work to be done here in terms of improving the delivery efficiency, potentially by identifying even more efficient cell-penetrating peptides. So I'll give a brief summary of what I talked about today. So first, I hope that I've convinced you that our system-delivered complementation in planta allows us to unambiguously study and quantify protein delivery in plants, and that it is a necessary system that gets around the limitations of standard co-localization analysis, which is constrained by the spatial resolution limitations of standard confocal microscopy. Next, what I hope I showed you is that plant homeoproteins or plant homeodomain proteins are generally cell-penetrating, which is an interesting bit of biology because it mirrors something which has been known in animals for a long time, but which has not been known in plants. Third, I hope that I showed you that the delivery efficiency of these peptides that we derive from homeoproteins is sequence dependent, and concentration dependent. Finally, that homeoprotein-based cell-penetrating peptides internalize into plant cells partially via endocytosis, and there's some interesting engineering that we can do in the future here. In animal systems, it's known that escaping from endosomes after endocytosis is very inefficient, and so there's future effort that we can conduct here in terms of trying to engineer endosomal escape from the endosome if endocytosis is indeed occurring. Finally, I hope what I showed you is that we can deliver functional cargoes with homeodomain-based cell-penetrating peptides. And going forward, that we're moving towards delivering more interesting, more functional protein cargoes. So moving forward, we're interested in a variety of different future projects. First, one that I'm really interested in is conducting the work that I've described in non-model species. So conducting this work in crop species. So moving to species like rice, barley, citrus, tomato, etc. We have some work ongoing to try to produce plants that express this reporter system, the DSIP reporter system, such that we can conduct experiments in crop species to evaluate if cell-penetrating peptides are also functional in these other species. Also moving forward, we're interested in delivering other functional protein cargoes, particularly in direct delivery of genome editors like Cas9, also attempting to deliver other functional transcription factors. Finally, more fundamentally, we're interested in further exploring the mechanism of internalization, particularly in understanding the role of the cell wall in how cell-penetrating peptides internalize into plants. Again, it's known in animals that there are a variety of different possible mechanisms, and it seems to be in plants that endocytosis is one of the mechanisms as well. However, we don't have a good understanding at all of how the cell wall plays with that. So with that, I will conclude and briefly give some acknowledgments. So first, thanks to Professor Marquita Landry for being my advisor for the last few years. We are fortunate to have some great lab staff who take care of our plants in the Landry Lab, including Ruby Nelson leading our team of lab managers. I've been fortunate to have some great mentors, including Jeff Wang and Dr. Nat Go. And I've also been fortunate to work with some great undergraduate students who have helped contribute to this work, Cerise, Menjay, Justin, Natalie, William. And I'd like to thank the Landry Lab in general, and then also thank the funding sources, thank the Department of Defense for funding the first half of my PhD, and also thank the Philomathea Foundation for funding the rest of my PhD. And thank you for your attention, and thanks to Professor Moon for putting this on. I'm happy to take any questions.
Fantastic. Thank you so much. It's very interesting. Also, I always love to see unexpected discovery, lead to new direction of the research. So that's fantastic. Let me ask Chris whether he has any question, comment on this talk.
I just, it's not my area really, but I just had a question on the CPPs. So are they used for drug delivery at all at the moment?
There are not many clinical applications of CPPs in humans. The challenge with CPPs in humans is that they lack cell specificity, and so they just generally internalize into all cells that you treat them with, which is a problem for clinical applications. They haven't found much clinical application. I would say in plants we care less about cell specificity, and so that's one of the reasons why we're interested in it. And the other technologies which do have some cell specificity in animals, they're likely too big to function in plants, because they tend to self-assemble into large constructs that are larger than the size exclusion limit of the cell wall.
I see. Yeah, that makes sense. And another question was, yeah, I'm not sure you really said why they penetrate the cell, the membrane. Is it just kind of charge distribution of the proteins, or what enables them to pass through these membranes?
Yeah, it's not entirely clear to us, especially in plants. In animals, there's a variety of different classes of cell-penetrating peptides. There's a class which are highly charged, highly positively charged, like R9, the model CPP that I talked about a little bit in my work. That one is highly charged, and the high charge density of it is part of the reason why it crosses cell membranes. But what's interesting is that an analogous peptide like lysine, which is also highly charged, does not cross the cell membrane. And so there's interesting nuances to why a cell penetrating peptide crosses, which are not entirely clear. The other challenge is the mechanism by which cell penetrating peptides cross. I kind of alluded to this a little bit in my work, is that the mechanism is dependent on the concentration of the peptide, what the peptide is attached to, and the type of peptide. So, for example, the peptides that I'm working with now, they appear to cross through a combination of endocytosis and something like membrane disruption. But there are other peptides that cross only via endocytosis. They don't. When endocytosis is disrupted, they don't cross the cell membrane at all. And so the mechanism by which it crosses the cell membrane is not entirely clear. When it is a energy independent disruption process, there are a variety of models that are proposed, but I don't think that there's good understanding of that. In plants, I would say there's no understanding of that, and so that needs to be an area of investigation in the future.
Cool. It seems like you're engineering this for biotechnology, but in the process, learning a lot about the interactions.
So, Chris, do you have any other question, comment?
No other questions or comments.
Great.
No.
So let me ask one question. Everything is clear. One thing I want to ask from more engineering perspective or regulatory perspective. I do understand the problem of GMOs because I'm working on those kind of problem for many years. One question I have is if you deliver protein into the plant, and then that protein happened to be genome editing, protein, the regulation is much less because, that you probably only, for example, Cas9, delivered, and you will probably also deliver the guide RNA somehow. And those things is not DNA, so regulatory body is much more generous. And then the modification happen within the, cell, so the regulation is much less. But other than gene editing application, the protein, the one downside is the stability, and that kind of stay temporarily and then just gone. So I just wondering what other application you could think of, when you're talking about delivery of the protein.
Yeah. So one application that we're also interested in is in a very niche application called plant regeneration. It's one of the steps in producing a transgenic plant. So let's say you wanted to make one that's not just gene edited, but transgenic. There's a step called regeneration, which is required to essentially take the plant from an immature state back to a mature full plant that can set seeds. And one of the applications we see there is transiently delivering a protein to encourage the plant to move from one developmental stage to another
I see.
to continue its development.
Got it.
And what we find there, the reason why we want it to be transient is that if that protein is continuously expressed, it interrupts the plant's normal development. You need it to kickstart the development, but then you want it to leave. And so that's one advantage of the transientness of protein delivery in that case. Another application that we are interested in, and this is a long-term stretch goal, I would say, we're not close to this, but one application that I've envisioned is there are times where when you know that there's going to be a stress to your plant in field. For example, you know a frost is coming, or you're expecting drought is coming, or you're expecting at this time of year this particular pest is known to infest plants. I could imagine a scenario in which you'd want to transiently deliver a transcription factor that prepares the plant for cold tolerance, for example, or for exposure to pests. But you don't want the plant to constantly be ready for cold exposure because then it stunts its growth. And so you could imagine transiently preparing the plant for cold exposure or for some other stressor. The plant survives that stress hypothetically, and then the protein doesn't remain long term though, and the plant goes back to normal function. That's another application that we envision. That's very out there, I'd say, at this point.
I see. I mean, as you probably already know, I mean, first GMO application is about that frost prevention, that's many decades ago. Now I see the 10 minutes after 11, so probably we're going to close and then probably chat afterward. Okay, so thank you all for joining and staying today. We'll meet again on July 17, Thursday, the same time, same Zoom link. We'll have Professor Timberg at University of Warwick and also Dr. Erin Garza at J. Craig Venter Institute. In fact, the 3 UK professor in a row, since last 2 weeks ago, I believe. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again, and I'll stop recording. Just 1 second.